Okay, so welcome everyone to this is now the fourth Easy Talk actually. Welcome everyone, we're really excited to have Earl Miller today as our fourth speaker. I don't think he needs a long introduction, but anyway, Dr. Miller received his PhD in 1990 from Princeton and spent the following 5 years as a postdoc at NIH. He's currently the Pekawa Professor of Neuroscience at Pekawa Institute for Learning and Memory at MIT. His work over the last years has produced numerous highly influential papers in the field of working memory, attention, decision-making, and learning, with a special focus on the role of prefrontal cortex. A recurring theme in Dr. Miller's research is how neuronal rhythms can shape cognition and behavior. Therefore, I find it quite fitting that today's talk is titled Cognition is Rhythm. So welcome, Earl, and I would say the screen is yours.
Thank you very much. It's a great pleasure to be here, and I thank you for the invitation. So let's jump right in. So what does our lab do? We study executive brain functions. Executive brain functions are the brain functions that do all the critical thinking when we think about high-level cognition: planning, focusing attention, learning and following rules, organizing thoughts. It involves functions like working memory, decision-making, attention, predictive coding. And the way we study executive brain functions is we record electrical signals, action potentials, local field potentials from the brains of non-human primates trained to perform cognitive demanding tasks. And we use computational modeling and analytics to try to make sense of these signals. And why do we do it? Well, we want to understand the brain. We want to understand cognition. That's all very cool and very fun. But we also want to understand cognitive dysfunction and diseases such as autism, schizophrenia, and attention deficit disorder, so we can open up pathways to new treatments for those diseases. Now, what are executive functions? Well, executive functions are the cognitive processes for selecting and monitoring behaviors that facilitate the attainment of goals. This requires the learning and use of top-down information, essentially the rules of the game, the goal-related knowledge of how our world works, what goals might be out there, what behaviors have been successful achieving those goals in the past, or what might be successful in the future. And this learning and use of top-down information is thought to be a major function of the prefrontal cortex, illustrated here in this very nice review by Berridge and Ernst. The prefrontal cortex is thought to absorb all this top-down information and use that to guide activity in the rest of the brain, so we can direct that activity towards attainment of goals. Now, over the years, we about 20 years ago, so we decided to begin to look at the role of the prefrontal cortex in absorbing top-down and using top-down information. And over the years, we've done a number of studies showing that the prefrontal cortex, if you train animals to do various tasks that require high-level top-down information, the prefrontal cortex absorbs and represents this information in a big way. All sorts of things that you need to make sense of the world and navigate a complex environment. So we taught animals things like monkeys to recognize shape categories, like cats versus dogs. When Andreas Nieder was in the laboratory, he trained monkeys to recognize numbers 1 through 5. In all these cases, we're asking our monkeys to recognize a cat or a dog or a number, not by focusing on what the things actually look like, the details of the sensory experience, but extract the essence, the things that make a stimulus, a cat or a dog, or correspond to a number 2 or number 3, regardless of how you present that number. And we've also studied the role of the prefrontal cortex in learning and representing high-level rules and principles, like same versus different, or up versus down. And I'll show you some other examples of that later in the talk. But the bottom line of all this work was that the prefrontal cortex seems to extract the goal-relevant structure of experience while discarding irrelevant details. So if you have the animal do things like recognize a number 4, regardless of how you present the number 4, what the prefrontal cortex represents is it responds to the anything with 4 items in it and throws away the details of how that you presented that number because that wasn't relevant to the task at hand. So the prefrontal cortex is only absorbing the information needed to solve the task at hand. Everything else is discarded. Now, when we first started doing this work about a little over 20 years ago now, we found something very curious. We found these effects. We would train a monkey to recognize cats versus dogs or small numbers or same versus different. And we saw these effects in 30 to 40% of randomly selected neurons in the prefrontal cortex. These are recordings with arrays of electrodes, so we weren't recording from single electrodes and single neurons and pre-selecting neurons for anything related to the task. We just randomly recorded from neurons in the prefrontal cortex and found that nearly half of them showed the effects I just described, all these high-level top-down information. Well, this is a problem because back, in the 20th century, when I was a graduate student, we used to think of the brain as kind of being like clockwork. Every neuron had one specific function, and neurons combine their signals from other neurons via physical connections. And that's what's illustrated here on the right. We have a bunch of units representing neurons back in primary visual cortex. They break down the visual scene into low-level features like small bars of light. Then with each stage of further processing, neurons at subsequent stages combine the signals of previous neurons at previous stages, and the response properties become more and more complicated until by the end you have neurons that will respond to things like complex shapes, or we have hundreds of millions of neurons in the brain. You go beyond that and you combine signals, and you have things like concepts like peace, love, and understanding. So this is the way we used to think of the brain. Every neuron had one specific function, and neurons combine signals because they have a physical connection. Well, then 34 to 40% of randomly selected neurons showing effect is a problem for this view of the brain, for this 20th century view of the brain, because the clockwork view of the brain posits that there should be a whole bunch of highly specialized neurons, all that do one thing. 40% of neurons doing one function doesn't make any sense at all. And when we first found these results, there was some skepticism, as there should be, because we're scientists, we're naturally skeptical. And we got explanations like, well, maybe the reason why 40% of your neurons are doing cats and dogs or numbers or same and different is because you spent so much time, spent weeks or months training the animals on this task, and maybe it's an abnormal situation. The task literally has taken over the brain. I never bought that explanation because our monkeys have like a couple months of experience solving these tasks, but we humans carry around decades of experience. I've been doing neuroscience for 36 years. I mean, like the kind of experience that we have is greatly far beyond anything monkeys have in performing these tasks. But I'm happy to say that through all the experimentation I showed you in the previous slide, and other people also started doing similar work, we found that the answer is that cortical neurons don't have a single function. They are multifunctional. They have something that we call mixed selectivity. In other words, they have different properties in different contexts. So if neurons really are multifunctional, this 30 to 40% is not a problem because that means that those neurons can be doing other things in other contexts. So this idea of multifunctional mixed selectivity neurons has gradually been accepted. I think now it's widely accepted. But I think the studies that really nailed it down is work by my colleagues Stefano Fusi and Mattia Rigatti using data that Melissa Warden collected in the lab. And they showed mathematically that this multifunctionality or mixed selectivity property is necessary for high-level cognition. It adds computational horsepower to the brain. It's like a neural bazaar where a wide range of information can interact. Information from all different sources can all intermingle in the same population of neurons. And that makes the brain smarter, allows the brain, by adding this, it adds a high-dimensional representational space that allows the brain to solve nonlinear problems instead of being limited to simple linear problems. And it increases storage capacity because if you distribute information storage among an overlapping population of neurons, you can essentially store an unlimited amount of information. And this Intermingling of information allows for faster learning because all the information is essentially there in the same, all this wide range of information is there in the same population of neurons, so information can be grabbed and put together really quickly. And for the same reason, it endows the brain with cognitive flexibility. So now that we know that mixed selectivity, multifunctionality is important for brain function, that's all well and fine, but mixed selectivity still is a bit of a problem if your view of the brain is a clock-like view of the brain. It doesn't fit with a clock-like view of the brain. In the, excuse me, old way we used to think about the brain in this clockwork paradigm, anatomy is destiny. If neurons have a physical connection, they'll activate one another, like we see here on this slide, where these neurons at subsequent stages are combining the activity from neurons at lower stages because they have a physical connection and because they have a their synapses are strong, have a strong weight. And the idea was that every perception, thought, and action has a unique network, something we call an ensemble. So the way we used to think of the brain is that if you have one concept, could be cat, or dog or whatever, you have one network, one ensemble that corresponds to that thought. And another thought would have a whole different ensemble, a whole different network that corresponds to that thought. Now you can develop associations. I can develop, I can learn that these two thoughts are related to one another and develop connections that will connect these two ensembles. But generally the way we used to think about the brain is that every perception, thought, and action had its own unique network or ensemble. But mixed selectivity implies that this is not the case. It isn't that every network has, every thought has its own ensemble. It's more like this, that mixed selectivity implies that ensembles are lying on top of ensembles, lying on top of ensembles. Ensembles are all mixed together and different ensembles share common elements. And I've illustrated that here by combining our blue ensemble and our red ensemble into one ensemble here, or one network here, where there's some units that are uniquely involved in one network or the other, one ensemble or the other. But some of these networks, some of these networks, some of these neurons are mixed selectivity neurons that are part of both networks. That's what mixed selectivity implies, that neurons are part of different ensembles. So if anatomy is destiny, how does that work? How does the brain select one ensemble from these overlapping connections? I mean, if anatomy was destiny and I wanted to activate the red ensemble here, well, the activity would run to the run to the blue ensemble by going through these mixed activity neurons. And pretty soon, if I try to activate the red network, the blue network also activates and our thoughts are a mush and a jumble. So how does how's the brain manage to select one ensemble from this overlapping anatomy? Well, one answer that we and others have come up with, mainly others, is that the networks are formed in the brain by synchronized oscillations. And you see that here, so in other words, cortical networks organized by doing the wave. The red ensemble synchronizes activity with other members of the red ensemble, and elements of the blue ensemble synchronize their activity with other elements of the blue ensemble. So in other words, cortical networks, can self-organize by this oscillatory organization. And that's illustrated here by this crowd doing the wave. Here we have a crowd, this wave is moving right to left across the crowd. So the crowd is self-organizing in this rhythmic fashion. So the idea is that neurons that hum together temporarily wire together. Now, I don't want to leave you the impression that we are the first ones to step down this path. We're merely following in the footsteps of pioneering work by people like Wolfsinger, Pascal Fries, Andreas Engel, and our colleagues who touched upon this theory many years ago. And I think that it's becoming increasingly clear that this synchronized oscillations do play a major role in organizing networks in the brain. And I'll point you to some brand new papers on this topic. Dimitris Panosis has shown that we can take this notion of neural activity forming, these synchronized oscillations forming networks, and bring it up even beyond the level of action potentials and LFPs, and look at the electrical fields generated by neural activity. And we see that same sort of representation and great representational stability there. And we also found that much like this crowd doing the wave, these oscillations in cortex actually form traveling waves. And hopefully that's something I'll have time to tell you about at the end of my talk. So the way to think about this is brain anatomy is not destiny, it's possibility. Brain anatomy is kind of like the road and highway system. It just says where traffic could go. Our thoughts and actions are where traffic actually flows from moment to moment. And the patterns of brainwave resonance, these patterns of synchronized oscillations, are what directs the traffic. Now, when Tim Bushman was in the lab, and he's now a professor at Princeton, he studied this directly by having animals switch back and forth between two rules. The monkeys were viewing colored-oriented bars. They could be vertical or horizontal, or they could be red and blue. And we cued the monkey from trial to trial, either pay attention to color or pay attention to orientation. And what Tim found is summarized here. This is the lateral prefrontal cortex, showing the lateral prefrontal cortex of a monkey. This is posterior, that's anterior. This is the principal sulcus. The monkey's eye would be about here looking forward. So it's like where on the side of my forehead, like right here is where this would be. Each of the circles shows a recording site. And the colored lines shows where we saw increase in synchrony between local field potentials when the animal was following either the pay attention to color rule or the pay attention orientation rule. Now, we looked across all frequencies from 1 to 100 hertz, and we found these effects in the beta range, about 12 to 30 hertz only. And as you can see here, when the animal is performing the color rule, one pattern of recording sites show these synchronized increase in the beta range. And when the animals find the orientation rule, you see an overlapping sites showing increased beta synchrony, but it's a unique pattern from the color rule. So we call these beta ensembles. They really seem to be helping form a representational structure, a network that corresponds to one rule or the other. And again, Demetrius Panosas has used things like dynamic causal modeling to take this work to an even higher level. So having found this result, we then started doing more experiments. We went back to our old data, and basically every experiment we've looked at where we've taught the animal top-down information like rules, categories, and concepts, we found these beta wave ensembles, different patterns of beta wave synchrony for cat versus dog, up versus down, choose same versus choose different. All the top-down information we could see at the single neuron level, we could also see in these beta wave, these beta oscillatory ensembles. So more recently, when Michael Lundqvist came to the lab, we began to look at the role of these oscillatory dynamics in the control of working memory. So here's Michael had a model for how working memory is controlled, and it was a computational model. He came to the lab to test it in the data. I'm happy to say our actual data supported all the major theories of the model, all the major features of the model. And what Michael found is shown here on a single trial. Here are local field potentials. Frequency is on the y-axis, time is on the x-axis. This is an experiment where the monkey had to hold two stimuli, two pictures in working memory. That's illustrated by S1 and S2. Here's where the first stimulus was presented. Here's where the second stimulus was presented. Here's the working memory delay. And the color shows the LFP power as a function of time. So what did Michael find? He found that sensory information in working memory, these two stimuli, the monkey that had the whole working memory, they were carried by bursts of spiking that were associated with bursts of gamma oscillations. And that's illustrated here. So the spiking carrying the working memories are co-occurring with bursts of gamma oscillations. There were also bursts of beta oscillations, as you see here, colored by the red circles. They were interspersed with the beta oscillation. The beta oscillations were interspersed with the gamma oscillations. In fact, we found that the beta and gamma bursts were anti-correlated. So that's what's shown here. Now, this was a single trial. Here's an average across trials. This is the burst rate for all the gamma bursting shown in the top curve, the reddish curve. And the brown curve shows the burst rate for the beta oscillations. And as you can see, they're mirror images of one another. gamma goes up along with spiking during encoding of stimuli in working memory. And it drops down in the beginning of the working memory delay, then gradually rises up over the course of the delay. And beta shows the exact opposite. It drops down when gamma goes up, and it goes up when gamma goes down. So beta and gamma are anti-correlated. Well, if the sensory information, the actual contents of working memory, is being carried by spiking and carried by gamma bursting associated with the spiking, what's beta doing? What's this anti-correlated beta doing? Well, you probably remember, because I just mentioned it a couple minutes ago, is that beta carries top-down information. And what that led us to speculate is that maybe this is how top-down control of working memory works. The most important thing about working memory isn't that you can hold stuff in mind, it's that you have volitional control over what you hold in mind. You can choose what to hold in working memory. You manipulate your thoughts in working memory. You choose when to stop thinking about things. Working memory is the main mechanism by which your brain wrests control from the environment and turns it into its own internal state. So this may provide a mechanism by which we get top-down control working memory. And the general idea is that beta is carrying the top-down control signals, and it has an inhibitory influence, a gating influence on gamma that's carrying the contents of working memory. So top-down information are the control signals by the brain carried by beta rhythms. They inhibit or gate the gamma rhythms carrying bottom information. Thereby, you can gate access to and clear out and manipulate the contents of working memory. Now, Michael Lundquist published a paper 4 years ago now, where he actually showed in great detail how these gamma and beta bursts can control access to working memory, can manipulate information in working memory, and can clear out the contents of working memory. So I point you to that paper if you want to know those details. But it clearly showed there's this gamma beta cycling dance that corresponds to control operations of working memory. When Andre Bastos and Roman Lunis were in the lab, they started to look more closely at these beta-gamma interactions by using laminar electrodes to record from all layers of cortex simultaneously. Now, why did they want to do that? They did that because we know from anatomy that the deep layers of cortex are the feedback layers that carry feedback signals from the front of the brain to the back of the brain. Whereas the superficial layers of cortex, they feed forward information, sensory information, from the back of the brain to the front of the brain. So if this hypothesis is correct, we would expect to see more top-down beta in the deep layers of cortex and more bottom-up gamma feeding forward sensory information in the superficial layers of cortex. And long story short, that's exactly what they found. In fact, what they found by not only studying these rhythms, but also studying their interactions, is summarized in this slide. What they found was that top-down information, our putative control signals, are carried by deep layer beta. Deep layer beta, in turn, regulates superficial layer beta. Superficial layer beta is weaker than deep layer beta, but it's there. And Granger's causality analysis showed that the deep layer beta was regulating superficial layer beta. And it also showed that superficial layer beta inhibits the gamma spiking carrying bottom-up information. So we only see gamma bursts illustrated here and spikes illustrated by these vertical lines. We only see them when beta is waning. So that's the idea, is that you have this cross layer, deep layer, superficial layer, beta gamma control content interaction that explains the top-down control of working memory. But then we start thinking, why does this only work for working memory and frontal cortex? In fact, you see these same dynamics, beta, gamma, anti-correlation. You see them all over the place in cortex. You see them in frontal cortex, parietal cortex, visual cortex. Michael Lundquist did a study where he recorded in all these cortical areas simultaneously and found these same push-pull beta-gamma interactions everywhere we looked. In fact, this is a well-known phenomenon in motor cortex. If you have subjects, if you tell them, get ready to press a button, but don't do it yet, you'll see beta, why they're inhibiting the response. The moment they get the go signal, beta drops, the gamma goes up, and they press the button. So these same dynamics beta controlling gamma, beta inhibiting gamma could play a role in cortical functioning in general. So this suggests the fact that they exist all over cortex suggests that similar mechanisms are at play all over cortex. Now, one thing I'll mention, just a little housekeeping here, is that Michael found in the study where he recorded from frontal, parietal, and visual cortex that all the frequency bands actually increase slightly in frequency as you go from the back of the brain to the front of the brain. So beta in frontal cortex is alpha in visual cortex. It just they're just the same function, just a slightly lower frequency. So alpha becomes in visual cortex becomes beta in frontal cortex as you move forward in the brain. So now I'll be talking about not just beta, but alpha and beta. Alpha being just below beta. Now, one thing we thought that this may play a role in is predictive coding. Predictive coding is something your cortex does all the time. The brain avoids sensory overload by constantly generating predictions of forthcoming sensory inputs. Your brain can't possibly process all the information flooding into your senses. So it makes these predictions, and predicted sensory inputs are suppressed. They're not informative. The brain saw them coming. The brain predicted them. They can't be that, they're not that informative. They're not interesting. However, unexpected inputs, known as prediction errors, are fed forward. The brain didn't see those coming. They weren't predicted, so they're fed forward, they're fully processed, and it updates our mental model of the world so we can make better predictions in the future. Now, our hypothesis, everybody agrees that predictive coding is something the brain does. In fact, it's a major theory of autism now, deficits in predictive coding. But no one really knows how the brain does it yet. One hypothesis is that there's specialized prediction circuits at every level of cortical processing that compute mismatches between predictions and sensory inputs, generates prediction errors. But we have a simpler hypothesis based on our alpha, beta, and gamma dynamics, namely that beta carries predictions, beta suppresses the gamma that would feed forward those predictions, and prediction errors are feeding forward by gamma, are the sensory signals fed forward by gamma that were unaffected by beta. So you don't need specialized circuits at every level of processing. You just need to send beta predictions via beta that target all the circuits in visual cortex or sensory cortex that would process a predicted input. Then the prediction errors are anything that hasn't been touched, that's been unaffected by beta predictions. So Andre Bastos, when he's in the lab, we did an experiment in collaboration with Nancy Coppell at Boston University, and we found evidence that supports this hypothesis. The animal was performing a working memory task where the objects the animal was holding a working memory were either unpredictable to the animal or they were highly predictable, like the same object multiple trials in a row. And what's shown here on this slide is the results of recordings from parietal cortex, area 7A, and area LIP, the frontal cortex, the frontal eye fields, and prefrontal cortex, and area V4. The blue shows the increase in coherence in alpha beta whenever the object was predictable, and gamma shows the coherence whenever an object was unpredictable, when it was a prediction error. The color indicates whether the synchrony was higher for predictable versus unpredictable in the case of prediction, blue, and the red shows prediction errors. The thickness of the line shows a degree of synchrony. So as you can see, for gamma, feed forward prediction errors dominate everything. It's pretty much all you see in gamma, feed forward prediction errors. Increased gamma coherence to unpredictable objects, I should say. And for alpha and beta, what we see is largely alpha, beta dominate whenever an object was predictable, whenever it was not a prediction error. So you can see that there's increased coherence between all these, between frontal eye fields and LIP, prefrontal cortex, V4. You see that in both alpha and beta when the stimulus was predictable. And you see an increase in gamma coherence whenever the stimulus was unpredictable and therefore a prediction error. The one exception is area 7A. And that's because work from Nancy Coppell's group shows that 7A is exceptional. The beta in 7A has a specific role in updating the contents of working memory. But generally, what we see is what was predicted by our model, and that is greater alpha beta coherence for predictable objects and increased gamma coherence for unpredictable objects. Notably, the gamma coherence was stronger in the feed-forward direction from visual cortex to parietal and frontal cortex, whereas the alpha-beta coherence was stronger in the feedback direction from frontal and parietal cortex back to visual cortex. And also notably, these effects were stimulus-specific. The effects were strongest at the recording sites, where the spiking at that recording site preferred that specific unpredicted or predicted stimulus. So these alpha-beta dynamics isn't just turning off a faucet that's turning on and off sensory processing in general. It can target specific stimuli, and the beta can suppress the processing of specific stimuli back in visual cortex, and the gamma can feed forward information about specific unpredicted stimuli. Okay, another set of experiments we've done recently is we've began to look at the role of general anesthesia in causing loss of consciousness. We're interested in cognition, so we thought it'd be interesting to know what happens when you lose cognition, lose consciousness under general anesthesia. This may be disturbing to know, but although we've used it, general anesthesia, for about over 100 years now, medical doctors don't really know why it works. They just know it does work. So this work is done in collaboration with Emory Brown's laboratory. Emory Brown is professor of neuroscience in our department, but also an anesthesiologist at Massachusetts General Hospital. So he's very interested in what causes unconsciousness and what we can do to make general anesthetic safer and more effective. So it used to be thought the folk wisdom was that anesthesia just kind of turns off your brain. Well, it doesn't just turn off your brain. What it does is it drastically changes the oscillatory dynamics of your brain. So here's recordings from 4 different cortical areas. STG is the superior temporal gyrus, auditory cortex. PPC is the posterior parietal cortex. Here's the prefrontal cortex. Here's frontal eye fields. These are LFP recordings. And here's spike recordings for about 20 seconds in time. So what happens when the animal's completely awake, we see a lot of high frequency chatter. This is because of communication through coherence, because of these patterns of oscillatory dynamics, forming networks, unforming networks, reestablishing networks. There's a lot of high frequency chatter going on because there's a lot of thoughts and processing going on in the awake cortex. But after we anesthetize the animals with a common anesthetic called propofol, you see something very different. All that high frequency chatter is replaced by a low frequency 1 hertz delta wave humming once a second. All the high frequency stuff we have associated with consciousness, gamma, alpha, beta, they're there, but they're in a severely weakened form. Instead, what's dominating the brain is this low frequency oscillatory dynamics, low frequency hum. And you can see from the spiking, this creates a bunch of up and down states with a period of 1 second. Up states with a bunch of spiking, followed by down states with no spiking. So your cortex is just like spiking, unspiking once a second when you're under anesthesia. I mean, this not only replaces the high frequency dynamics that we associate with consciousness, but it also presumably prevents different areas of cortex from communicating with one another effectively, thereby rendering unconsciousness. Now, having known this, we decided that what we would do is maybe we could actually restore consciousness in animals that were unconscious due to propofol. Maybe we could restore consciousness by replacing these low frequency dynamics with high frequency dynamics through electrical stimulation of the brain. So we chose the thalamus because it's thought the thalamus forms these loops with cortex, and it's thought that the high frequency coordination in cortex depends on these low frequency signals fed to the cortex by the thalamus. In fact, one major theory of consciousness targets these or focuses on these thalamal cortical loops. So what we did is we use high frequency electrical stimulation to stimulate the anterior thalamus to try to restore consciousness in the animals, and it worked. When you stimulate the thalamus at high frequency, what happens is the cortex goes from this low frequency hum, it reverts back to the high frequency chatter, and the animals are completely unconscious. And you start stimulating the thalamus, and they open their eyes, they sit up, they start moving around, they start responding to sensory stimulation. We wake them up even though they're still under the influence of propofol. Then we turn off the thalamic stimulation, and they fall back under unconsciousness again. And we're using studies like these to try to develop safer methods of delivering anesthesia, ways of inducing unconsciousness that are more targeted, that are therefore safer for patients, and also ways of monitoring anesthesia state so we don't over-deliver anesthesia to patients, which is a real problem in young patients and older patients, because if they receive too much anesthesia, you often get long-lasting cognitive deficits. So we're trying to use these data and further experiments to make anesthesia safer. Now, another thing I'll tell you about is, so far I've talked about, at the beginning of the working memory section of this talk, I talked about these complex dynamics where we have these bursts of gamma and bursts of beta interspersed with one another. But after that, I started talking about beta and gamma in a more general way. But given that on single trials, this bursty activity is really quite complex and seems to have this dynamic dance, we've got to look closer at these dynamics. And I'll just tell you briefly about a brand new paper that we just published in the past month or so, and that shows that we show that low frequency oscillations, everything below 30 hertz, forms traveling waves in cortex. So here's a phase map of a beta wave over time. This is the phases shown in red. This is the prefrontal cortex. The recording wave was right here. And this is the same recording array just spread out over time. And you can see that the phase map changes as a function of time, so that a wave moves from dorsal posterior part of the array and sweeps down to the anterior ventral part of the array. And here's a couple real-time examples from single recording arrays from a single session of these oscillatory waves moving across the prefrontal cortex. You can also see that here's a map of the recording sites on the array over time, and you can see this orderly phase progression indicative of a wave moving across the cortex. So what happens? We found that traveling waves in the prefrontal cortex during a working memory task, the traveling waves are actually there all the time. But when the animal's not doing a task, they move in different directions, they go back and forth, they rotate around, with no order to them. Now, some of these waves are planar, they move straight across the cortex, but a lot of them rotate around anatomical points in the prefrontal cortex. And interestingly, it isn't just one phenomenon where all the waves are moving in the same direction. Waves in different frequency bands often travel in different directions at the same time. So there's a lot of complexity here. And we found they change direction with task performance. They start out kind of moving in a seemingly random kind of back and forth kind of way. There's no order to them. And the moment the monkey starts a task, they become highly directional and stereotyped. Then when the monkey finishes the task, they go back to their baseline, kind of moving around in any direction they want. And what we found just recently, just published this week, is that propofol anesthesia doesn't just alter the dynamics, the power of oscillatory dynamics in the brain. It doesn't just increase low frequency oscillatory power, it also drastically alters the travel of traveling waves. It takes all these traveling waves you see in the awake state and makes them all move in different directions than they do in the awake state. And finally, just the last couple minutes, I'll show you one last role for alpha and beta and gamma, and that is in transferring information between the right and left brain, between the right and left hemispheres. Now, we all know, probably we all know that vision is split down the middle. The right half of your visual field, your right visual field goes to the left half of your brain and vice versa. And it used to be thought that, and this is true back in visual cortex, it's been known since I was a graduate student, I've known before I was a graduate student, known for a long time. But it used to be thought that somehow this gets right and left sides of visual space get united together in higher level cortex, in the prefrontal cortex. But we know from work in our laboratory that Tim Bushman, Dimitris Panosis, and Simon Kornbluth did, is that even in prefrontal cortex, even in working memory, there's a split in vision down the middle. The right and left sides of vision have independent working memory capacities, as if we are all a bunch of split brain patients. We found this in monkeys, but working with Ed Vogel at University of Chicago, we found the same thing in humans. In lots of human studies. So everybody, visually speaking, everybody's kind of like a split brain patient where the right and left halves of visual space are independent of one another. But visual cognition seems seamless. So how does the brain unite right and left sides of vision? This is something we're studying now in the lab. And our first experiment on this, what we decided to do is have the animal switch a working memory between the right brain and the left brain, or between the left brain and right brain, either direction. What we did is we had the animal fixate a spot of light on one side of a computer screen. We presented a to be remembered object, a picture like this banana, to one side of gaze. So since the picture was presented on the right side, that meant that it would be processed and held in the left hemisphere. Then in the middle of the working memory delay, on one half of the trials, we had the animal move its eye from one side of the screen to the other. Now, if that stimulus was still on the screen, that would change the hemisphere by which the stimulus is processed from one side of the brain to the other. But the stimulus is not on the screen. The animal is just holding in a working memory. And what we found is that the memory traces disappeared from one side of the brain and then reappeared on the other side of the brain. So the working memory traces, as a result of this eye movement, changing gaze, moved the working memory from one side of the brain to the other. So the working memory would stay in register where the eye was now pointing. So for that particular result, I point you to our paper from last year. But what we found is that during this transfer of working memories from one side of the brain to the other, we saw the same push-pull beta gamma dynamics. I haven't talked much about theta yet. Theta is not very strong in cortex, but it cross-frequency couples with gamma. So theta and gamma are kind of one thing. And then in between that is beta, which is the inhibitory control signal. So here is the frequency on the y-axis. Now what we're measuring here is coherence in local field potentials between the right brain and the left brain. And this is at a function of time. Here's the saccade, that when the saccade occurred, that meant the working memory was going to be transferred from one side of the brain to the other. And what happens when the animal makes that saccade? First thing to happen is that beta decreases. That decrease in beta allows gamma to be beta coherence between the right and left brain decreases. As a result, gamma increases, gamma coherence between the right and left brain increases, and the working memory is transferred. So this suggests that the way the working memory gets transferred between the right and left brain is you got to decrease beta. That allows gamma to synchronize between the right and left brain, and then the working memory gets transferred. And in support of that, when we did Granger's causality analysis, we found that the direction of influence, the direction of synchrony, was always in the direction of the sender to the receiver. If the working memory was moving from left to right, that was the direction of the synchrony. If the working memory was moving from right to left, that was the direction of the synchrony. So the direction of the synchrony followed the direction of the working memory transfer. Okay, so to sum up, we argue that alpha beta is like a control signal that feeds back top-down information through cortex. It regulates the expression of gamma that traffics bottom-up sensory information by spikes associated with gamma, and that's the contents of sensory experience and working memory. And we think this is a modal cortical circuit. This balance, this interaction between alpha and beta and gamma is a modal cortical circuit that plays a general role in allowing the brain to exert top-down control over sensory processing, attention, working memory, predictive coding, motor output, and even the transfer of memories between the cerebral hemispheres. So with that, I thank you for your attention, and I thank you for the opportunity to tell you about our work. And most importantly, I thank all the students and postdocs in my lab who really do all the hard work in making these discoveries happen. Thank you.
Thank you very much for the very nice talk. You